My name is Claire Gizowski, and I'm currently a postdoctoral fellow in Zachary Knight's lab at the University of California in San Francisco. Previously, I was a PhD student in Charles Bourque's lab at McGill University. And the paper that I want to tell you about in this video concerns the last part of my work as a PhD student at McGill, which was recently published in Nature. So the suprachiasmatic nucleus, or SCN for short, serves as the body's master circadian clock, which adaptively coordinates changes in physiology and in behavior in anticipation of changing requirements throughout the 24-hour day-night cycle. So for instance, the SCN opposes overnight dehydration by lowering body temperature to reduce water loss during sleep. This response is thought to limit evaporative water loss from the airway tract by reducing the thermal gradient between the organism and its environment. It's also been shown that in humans, following a salt load, that their body temperature also decreases. So responses such as these can also be driven by central brain regions that detect changes in osmolality and sodium concentration to oppose unscheduled rises in osmolality in mice to maintain homeostasis. We therefore wondered whether these osmosodium sensors, and in this case specifically the OVLT, require clock output networks to drive homeostatic responses. So it's thought that the high activity of SCN vasopressin neurons, or SCN VP neurons, during the light or the sleep period is what drives a reduction in core body temperature during sleep. We therefore wondered whether a salt load given during the active period or the dark period, when these neurons are thought to be silent, could activate these neurons and drive a decrease in core body temperature. So to examine the effects of systemic salt on core body temperature and the underlying thermoregulatory mechanisms that may be contributing to the associated changes in core body temperature, we used infrared thermography as a non-invasive approach to investigate this question. So using IRT, we measured temperature changes from the eye, which we use as a measure for core body temperature, and from the brown adipose tissue. So the bat is thought to be one of the main sources that regulates non-shivering thermogenesis. And in these videos shown here, this is the same mouse, but in two different trials. And as you can see, following the isotonic saline injection, during the dark period at a time during which SCNVP neuron activity is low, there is no visible change in either bat or eye temperature. However, following a hypertonic saline injection in this bottom panel here, you can see that there's a very visible reduction in the temperature of the bat and of the eye. So we can prevent SCNVP neuron activation using a chemogenetic approach during the hypertonic saline injection. And when we prevent the activation of SCNVP neurons, this reduces the effects of salt on core body temperature and on bat temperature. However, we can also mimic the effects of hypertonic saline on core body temperature and bat temperature by optogenetically stimulating SCNVP neurons. So using an array of viral retrograde tracing techniques as well as electrophysiological techniques in vitro, we were able to determine that osmosodium sensing OVLT GABA neurons directly project to SCN vasopressin neurons. We wanted to confirm that this pathway in vivo is what drives the decrease in core body temperature and the decrease in bat temperature, as seen following the hypertonic saline injection. So using optogenetics, we specifically activated the axon terminals of OVLT GABA neurons within the SCN, and we found that this effect mimics the effects of salt on core body temperature and on bat temperature by an excitatory effect of GABA on SCNVP neurons during the dark period. This work would not have been possible without the generous funding from the Canadian Institutes of Health Research, and this work was done at McGill University and part of the McGill University Health Center.